Hello friends, welcome to Light Classes. If you are watching my video, please like and subscribe my channel. In previous video, we have seen the history or the discovery of mitochondria. Now in this video, we are going to see the structure of mitochondria. Here you can see the structure of mitochondria. This is outer membrane which is more permeable and this is inner membrane which is less permeable. Here you can see the round structure in which thread-like structure is present. This is a DNA. And here you can see the fluid part, means liquid part, it is known as matrix. Here you can see some structure inside the inner membrane, on the surface of inner membrane, that is oxysome, or we can say it as a F1 particle. Here you can see the ribosome, it is of 70S type, and here perimitochondrial space is present. It is about 100 angstrom unit. Here you can see it is a perimitochondrial space which is about 100 angstrom unit. Now if we want to see this particle, then we are going to see the structure. You can see here the structure of F1 particle or oxysome. We can call it as a oxysome or F1 particle. This is head, which is known as F1. It is stalk. Here it is a base F0. Here ATP synthetase enzymes are present which synthesize ATP. ETC cycle takes place in this oxysome particle or F1 particle. ETC means electron transport chain. This cycle takes place inside the oxysome. So this was the structure of mitochondria. Now we are going to see the description about mitochondria. Mitochondria is double membrane bound organelle. You can see outer membrane and inner membrane. Two membrane, outer membrane is permeable and it is smooth, while the inner membrane is less permeable and folded at various places. Means here you can see the folds. Here you can see the folds. And these folds are known as cisternae or a palade. Cisternae contains number of oxysome particles. These oxysome particles are also known as F1 particle, Moron particles or elementary particles because these particles are observed or discovered by F. Moron. Name of scientist is F. Moron. He observed these particles. So these particles are also known as F. Moron or Moron particles, or we can call it as F1 or F0 particles. So now we are going to see what is matrix and what is F1 particles and how this works, the function of mitochondria. First we will write some points in description, okay? This mitochondria is double bounded membrane or double membrane bound organelle. Double membrane bound organelle, okay? Outer membrane, now double membrane, so outer membrane, more permeable and smooth, while inner membrane is less permeable and rough, means they are having a folds. We will write rough or having folds. These folds are known as cisternae. The folded structure is cisternae. or we can say it as a pallid. This cisternae consists of number of oxysome particles on the surface, inner surface. Consists of number of F1 particles or oxysomes. Okay, number of F1 particles or oxysomes. Now we are going to write here, these oxysome particles are known as oxysomes, known as first F1 particle, F0 particle, Moron particle, because discovered or observed by Moron, F Moron, and also elementary particles. Elementary particles, okay. So oxysome known as F1 particle, F0 particle, Moron particle or elementary particles. Now we are going to see what is F1 particles. If we see the F1 particle structure, then F1 is the head, it continues etc cycle, okay. F1 is head and continue etc cycle, okay. Now base F0, base F0, it is a factor by which oxysome is attached to the inner membrane. F0 is the factor to which oxysome attached to inner membrane, okay, attached to inner membrane. Means we can say it as a OSCP. OSCP means what? Oligomycin sensitivity conforming proteins. These are the type of proteins. Means these proteins are helpful to oxysome attached on the inner membrane of mitochondria. What is OSCP, okay? Means the full form of OSCP is oligomycin sensitivity conforming protein. These proteins are helpful to attach oxysome on the inner membrane of the mitochondria. Okay. Now we will see the number of oxysome present in the mitochondria. Number of oxysome in mitochondria is the number of oxysome in mitochondria is 10 raised to 4. Number of oxysome in each mitochondria is 10 raised to 4 to 10 raised to 5. And now we are talking about the matrix. Matrix is the fluid present inside the mitochondria, or we can say that liquid part present in cell organelles known as matrix. Now this matrix consists of, means mitochondrial matrix consists of all enzymes of Krebs cycle. Krebs cycle occurs inside the matrix of mitochondria. Means we will write here, it consists of all enzymes of Krebs cycle. And this Krebs cycle takes place or occurs inside the matrix of mitochondria. Right here, Krebs cycle occurs in the matrix of mitochondria. Matrix of mitochondria. Now, this mitochondria consists of many enzymes which are useful in the Krebs cycle, such as we will take an example, succinic dehydrogenase enzyme. Because this enzyme is attached to the inner membrane of the mitochondria. Right here in bracket, succinic dehydrogenase enzyme is attached to inner membrane of mitochondria. Okay. Now we will talk about DNA. We have seen in the structure, the rounded structure in which the thread-like structure is present. It is a DNA. Now we will see the DNA of mitochondria. Okay. DNA. Okay. DNA is a circular structure present in 2 to 10 copies. Okay. It is a circular structure. Present inside the matrix in 2 to 10 copies. Now, it is a circular structure present inside the matrix and it is 2 to 10 copies inside the mitochondria. Mitochondria is only the cell organelle which consists own DNA. And so, this DNA is helpful to replicate or to replicate the energy inside the mitochondria. Means mitochondria have power of replication. Mitochondria have power of replication. Each mitochondria arises from pre-existing mitochondria. As we have seen, the lifespan of mitochondria is 5 to 10 days. Then this new mitochondria exist from the pre-existing mitochondria or arise from pre-existing mitochondria, and the old mitochondria is engulfed by lysosome. So we will write here, each mitochondria arise from pre-existing mitochondria. Okay. Now we will see, we have seen about the matrix and DNA of mitochondria. Now we are going to see the ribosome of mitochondria. If we talk about the ribosome inside the mitochondria is of 70S type. 70S type ribosome. Own replication power. This ribosome have own replication power. Mitochondria can synthesize own proteins. This mitochondria can synthesize own protein because of 70S type of ribosome. Own replication power. Okay. Ribosomes are of 70S type in mitochondria, and they also have own replication power. They can replicate their self. And mitochondria also synthesize own proteins due to ribosomes. Due to presence of ribosome, they can synthesize own proteins. Okay. Now, mitochondria is also known as semi-autonomous organ. Mitochondria is also known as semi-autonomous organelle. Is semi-autonomous organelle. This Morgellus scientist said that the mitochondria is the prokaryotic cell which lives symbiotically in eukaryotic cell. Morgellus scientist said that mitochondria are prokaryotic cell which lives symbiotically in eukaryotic cell. Now, mitochondria is the only cell which having DNA inside it. Means other cell organelles such as lysosomes, Golgi body or other endoplasmic reticulum, they do not have their own another DNA, but this mitochondria is the only cell organelle which consists own DNA. So mitochondria is a cell within the cell organelle. So we will write here mitochondria is cell within cell organelle. Okay. The group of mitochondria in cell is known as chondriome. Group of mitochondria in cell called as, these are important for NEET and CET syllabus. Okay, the group of mitochondria is called as chondriome, and the group of mitochondria, here we have written the group of mitochondria is called the chondriome, and the another term is chondriosphere. Chondriosphere detects the disease, means in scurvy disease all mitochondria are fused together due to deficiency of vitamins and bleeding gums. Okay, what is chondriosphere? We will write here in scurvy disease. in scurvy disease. All mitochondria are fused together and why these mitochondria are fused together? Because deficiency of vitamin and bleeding gums. We will write here scurvy disease. Okay, in bracket we will write deficiency of vitamin C, mostly vitamin C. Vitamin C and bleeding gums. Now, in last topic we are just going to say about two scientists, that is Hershey and Margit, who discovered mitochondrial DNA. These two scientists discovered the mitochondrial DNA. We have talked about many scientists that they have given another terms to mitochondria, but Hershey and Margit are the two scientists who discovered the mitochondrial DNA. Scientist Hershey and Margit, who observed or discovered mitochondrial DNA. Okay, so this was the structure and the description about the mitochondria. In next video we are going to see the functions of mitochondria. So thank you friends for watching my video. Thanks.